Good afternoon, everyone. Thank you for joining this session. I would like to introduce myself, Emma Peel, and my colleagues, Dr. Luke Silver and Dr. Elle McLennan. We are all postdocs in the Australasian Wildlife Genomics Group at the University of Sydney, and today it is our pleasure to chair this session, highlighting the research of early career researchers who work in biodiversity genomics. How the session will run today is that we will play back each talk, and then each talk will be followed by a Q&A session, and a reminder to please submit all your questions via the Slido, which we will put to the speaker after each talk. So our first speaker today is Liam Barry. Liam is a senior technician in forensic biology at the Institute of Environmental Science and Research in Auckland, New Zealand. He's currently working in the field of human forensics, but his research has focused on species identification in non-human forensic science. Liam will discuss his Master of Science project on plant and animal species identification, which was carried out in collaboration with the University of Auckland. So today, the title of Liam's talk is Forensic Universal Animal and Plant Identification.
Welcome everyone. Thanks for taking the time to listen to my talk. I'll be discussing a forensic application of biodiversity genomics that's ongoing at the Institute of Environmental Science and Research, or ESR. I'd like to acknowledge some of my colleagues that are active in this area and that have contributed to what I'll discuss today, that are listed here on the slide, but particularly Olivia Jugovic, Dr. Ryan England, Dr. Shane Starrick, Janet Stacey, and Dr. Sally Ann Harbison, who were my supervisors and who helped me with the bioinformatics side of this project. So I want to start by addressing the question, what does forensic science have to do with biodiversity genomics? So forensic science might conjure up images of yellow crime scene tape or fingerprint dusting, and that's correct, that's what forensic science does, but at its most basic, forensic science is the application of scientific expertise to answer or inform legal questions. So as I'm sure you'll appreciate, that's a very broad scope, and it allows the forensic scientist a lot of leeway to focus on a lot of different questions. So it's quite ironic that forensic biology has traditionally been very narrowly focused on just one species out of millions in existence, which is obviously humans. But some forensic scientists have also dedicated their time and effort into considering non-human genomes as well, and that's the field of wildlife forensics. So what legal questions does wildlife forensics consider? And what's the legal basis for wildlife forensics? A lot of it comes back to the Convention of International Trade in Endangered Species of Wild Flora and Fauna, also known as CITES, which is a large international treaty that aims to ensure international trade in specimens of wild plants and animals does not threaten the survival of these species. So it provides the basis for the legal frameworks of hundreds of countries worldwide with respect to wildlife trafficking, and 184 states are parties to this convention. And forensic science is useful because these parties are all obliged to enforce the restrictions in wildlife trade, particularly for highly processed samples where morphological analysis is not possible, and that includes traditional medicine samples, which this research focused on. So this is obviously a very wide-reaching treaty. 184 countries that are highlighted in green here are parties to it, and the illegal wildlife trade is also a very, very large illegal trade worldwide. According to the 2024 UN Office on Drugs and Crime World Wildlife Report, between 2015 and 2021, 16,000 tons of trafficked material was seized across 162 countries. And the vast majority of these species that were seized were listed in the CITES appendices. So from the Earth Biogenome Project, their grand vision is to create a new foundation for biology to drive solutions for preserving biodiversity and sustaining human societies. And I believe the work we've done in wildlife forensics at ESR fits into this vision. And I know what we've done depends very heavily on the work of many researchers in biodiversity genomics. So I hope I've explained how forensic science and biodiversity genomics are aligned, specifically with regards to the CITES agreement and their shared goals. So I now move on to this work that we've done at ESR. One of the major goals of the project that I was involved with was the development of a universal species identification method. So that's what I undertook for my master's thesis. And I focused, as I mentioned, on traditional medicine samples. So the diversity that's present not only in the physical nature of these samples, but also in the range of species necessitates a very broad method. So design choices in both the laboratory and in bioinformatics were geared to maximize our ability to gain useful information from the wide variability that we saw. So for example, the method begins with a general purpose extraction method that was selected out of 3 different commercial kits. 3 universal animal markers and 2 universal plant markers were selected from the literature and combined into 2 different multiplexers. These PCR amplicons are then sequenced on the Illumina-MiSeq, which allows the application of the method to mixed samples. And to genomics researchers, I imagine the Illumina-MiSeq might be a technology that's been around for a long time, but in forensic science, even the MiSeq has not been widely validated and accepted into routine casework. And the difficulty in validating new methods is one of the major reasons why a universal method was the goal of this research, because it's not realistic to validate hundreds or thousands of different assays for potentially tens or hundreds of thousands of species of interest. So after sequencing, sequence files are processed and compared to reference databases by blast searching, and these are checked. Blast results are checked for quality. Currently, the NCBI nucleotide database is our reference database, but it presents a lot of difficulties that I'll talk about more later in the talk. And presentation of the results must also conform to the requirements of the courts. And as I mentioned, wildlife forensics is not as well studied compared to human forensics. So this reporting is not routine in many jurisdictions, including New Zealand. So moving on to some results. Method was shown to be very sensitive, with generating the correct result from as little as 1 picogram of input DNA into amplification. And this was using test ham jerky DNA that was store-bought for testing the method. And I'll touch on some of the other features of this plot later, including some of the erroneous database sequences that are seen in the cytochrome B and 16S markers. That relates to some issues with the reference database that I mentioned in the last slide. So similarly, for the plant multiplex, there was correct results to the genus level produced in both markers from very low input DNA amounts. The robustness of the multiplexes to non-target DNA was also assessed. It was found to be robust to mixtures of 1 in 200 parts of target to non-target DNA. But when there's no target DNA present, some microbial results can be generated, which we'll see more in the next slide. And following on from this testing, we carried out a survey of traditional medicine samples that were purchased from online stores in New Zealand, and also some that were donated by New Zealand CITES Management Authority. And this is an example of one of the results from one of these samples that was seized at the border by New Zealand CITES Management Authority, the Department of Conservation. And this was suspected of having tokay gecko, which is a CITES listed species. And in fact, you can see that this is present in the cytochrome oxidase marker, so in the middle graph, and also in a low level in the 16S marker on the left. And also present is Paludiscus sinensis, which is the Chinese soft-shell turtle. And there were also a lot of indications of other species, including some fungal species, particularly in the cytochrome oxidase marker. So reporting some of these lower coverage results that maybe were only present in one marker. As I mentioned previously, this was a key consideration from a forensic point of view. And imagining if these were casework samples led us to develop and implement some interpretation thresholds to help us interpret some of these lower results, to assign and justify the appropriate evidentiary weight. So these are the Chinese soft-shell turtle and the tokay gecko that we detected from the sample. And detecting this CITES listed gecko was a significant milestone in this research, and really emphasized the importance of doing this type of work and protecting these species. For interest in a different sample, we also detected the Saiga antelope, which was also CITES listed. So these results from the traditional medicine survey showed that this method is very successful in identifying a range of species from a range of different sample types, and previously shown also highly sensitive and robust to non-target DNA. And as we've shown in this traditional medicine, able to detect multiple different species from one sample. However, there is still plenty of progress to be made. So I'll just highlight some of the ongoing areas of opportunity and challenges that we're still looking at. So as I've already mentioned, databases, NCBI nucleotide is not a perfect database, has been well characterized to have erroneously annotated sequences, and that's been seen multiple times in the work we've done. So as I showed earlier, cytochrome B, there's a related species, Potomocheirus levatus, which is bush pig, also a member of the pig family, and there's also N16S, an insect species, and these are two erroneously labeled database sequences that we see consistently when we sequence pig DNA. But some of the other options that we've considered also have their own drawbacks. So BOLD also has a limited number of markers, which rules it out for consideration for the next generation of barcoding technology called super barcodes, which I'll touch on shortly. And other smaller databases often lack the range of species required for a universal method. In the future, particularly relevant from a biodiversity genomics point of view, are the limitations of barcoding as an identification method. So in this figure, hypothetical species A and species B overlap in their sequence diversity. So in this marker, they wouldn't be able to be differentiated using barcoding. Whereas species C is distinct from these two and would be able to be differentiated. But In the future, it's likely that a super barcoding method will be taken up. So this is sequencing entire mitogenomes or chloroplast genomes to increase the discriminatory power of the sequence information gained. And that's kind of the next evolution of where forensic species identification is going, that I think will bring it more in line with biodiversity genomics and show even more overlap between these two fields. Another difficulty, I mentioned success from a range of sample types, but not all sample types were equally successful. So, for example, the liquids and oil samples were a lot less successful in extracting and amplifying DNA, and that's kind of an ongoing consideration that we're working to resolve. And I'll lastly also mention, with regards to databases, we're also considering indigenous data sovereignty. So with traditional medicine samples, by definition, almost all of them have cultural significance, which must be taken into account when dealing with genetic data from these species and from these samples. And the global scale of the illegal wildlife trade means that indigenous cultures around the world need to be considered when assembling a database for use in this type of enforcement of the CITES Treaty. And that's something we're committed to at ESR, and we want to implement in a future wildlife forensics database. So that's all I had. Please direct any feedback to me at this email address, and thank you again for listening.
All right, thanks, Liam, for that talk. We'll just move straight on to our next talk, which is from Dr. Lara Parada, and she is a molecular marine ecologist with expertise in biodiversity genomics and aquatic ecosystem research. And Lara is currently a postdoctoral research associate as part of the Minderoo Oceanomics Centre at the University of Western Australia. And her current research is focused on using whole genome sequencing to further understand genome evolution in marine ray fin fishes, and she's previously worked across freshwater and marine systems. Dr. Parada has a keen interest in integrating biodiversity genomics into ecological research to advance finfish conservation. And today she's going to talk to us about genome evolution in marine ray fin fishes.
So hi everyone, thanks for dialing in today. My name's Lara and I'm going to be talking about genome evolution in marine ray fin fishes. So just before we get started, I'd like to start by acknowledging that I'm based in Perth and here at the University of Western Australia, we're situated on Whadjuk Noongar land, and the Whadjuk Noongar people remain the spiritual and cultural custodians of this land and continue to practice their values, languages, beliefs, and knowledge. So as sequencing technologies have advanced and sequencing has become more affordable, we've seen a real uptick in the number of reference genomes becoming available across the tree of life. This data is coming in the form of high-quality reference genomes, meeting the Earth Biogenome Project and Vertebrate Genome Project quality metrics. And for some samples, when the quality is not sufficient from the sample to generate this kind of data, we're still generating what many are referring to as high coverage or draft genomes. Whilst these draft genomes might not be EPP or VGP standard, they are still often a significant improvement on the sequencing data previously available. This increasing in data availability is opening up new possibilities in evolutionary studies, and it's providing the resolution required to answer questions in a new detail that was previously unattainable with barcoding and metagenomics. So one of the clades with the highest potential for understanding genomic evolution and ecological adaptation is fishes. And as you can see here, there are over 32,000 species of marine fish, or fishes. Let me start that again. One of the clades with the highest potential for understanding genomic evolution and ecological adaptation is fishes. As you can see here, there are over 32,000 species of fish, compared to less than 10,000 for most vertebrate clades. Even more significantly is that around 90% of fish are ray fin fishes, with much smaller numbers of sharks, rays, and other jawed fishes. So fish are an extremely diverse group inhabiting almost all water bodies on Earth. If we focus on marine species like I have here, you can see that they get a huge range of colors, morphologies, and sizes, each inhabiting a unique niche within its ecosystem. This diversification provides an ideal system for studying genomic evolution and the genomic drivers behind species diversity, ecological adaptation, and ecological success. So with these questions in mind and using marine fishes as a study organism, this project sought to improve our understanding on the variability of marine genomes. With these questions in mind, using marine fish as an example, this project sought to improve our understanding on the variability of marine ray-fin fish genomes. We wanted to delve into the relationship between phylogeny, ecology, and conservation metrics such as vulnerability and inbreeding coefficients, and try to discern whether species diversity is correlated with gene family diversification. So to answer these questions, we first had to acquire the genome assemblies of marine fish. To do this, we looked on NCBI for ray fin fish genomic data and filtered this down to only include whole genome sequencing. We then further filtered to only include things with Illumina technologies. Whilst this may introduce a slight bias in quality, we're currently working to filter out the poor quality genomes and develop a similar study using VGP quality reference genomes. So this decision to use short-read genome assemblies and exclude genome assemblies that incorporate long-read technologies made the compute a little bit more attainable, and it also ensures that there was no sequencing bias when we compared the downloaded genomes from NCBI to those that we're generating en masse in-house. So as part of the Minderoo Oceanomics Centre at the University of Western Australia, we're aiming to generate reference genomes for all marine vertebrates. However, when the sampling quality isn't sufficient for long-read genome technologies, we're generating high-coverage short-read genomes. So far, we've generated data for 426 fishes, resulting in a fairly comprehensive data set of 557 species fish for this study. Within this data set, it is important to note that there are a few duplicate species, and as previously mentioned, we're still working to quality check all of the genomes that we acquired from NCBI, so the data herein is all preliminary. So the first thing we wanted to do was look at how many genomes we had for each of the orders of fish. So along the x-axis here, you can see each of the fish orders, and just a count along the y-axis. The gray bars represent how many species there are in that order of fish, and the blue-gray bars represent how many genomes are available for that order. So as you can see, the gray bars are still significantly higher than the green ones, so we still have a lot of genomic sequencing to go. So the first main question of the study was how variable are marine fish genomes? To do this, we first checked the taxonomic coverage of our genomes. So whilst this plot is a little bit busy, the main takeaway is that the orders you see listed in blue and green are clades that we have data available for, and anything in grey was either missing or we excluded from the data set. For example, maybe they're all freshwater species. So this tree was downloaded from the Fish Tree of Life, and as you can see, the genome availability that we have is pretty well spread across the entire circle. The next thing we looked at was the genome size. So here along the x-axis, we have genome size in megabases compared to contig N50 on the y-axis. You can see that most of the marine fish genomes come in between the 500, 1000 megabase mark, and a lot of the contig N50s are also quite consistent, although they are low. 25,000 is not a very high number for contig N50, but it is what we would expect from short read assemblies. So before we get into the content of this plot, I just want to orient everyone because we've got a few like this coming up. So along the y-axis, we have all of our fish orders, and along the x-axis is the metrics that we're comparing. The straight line up and down the middle represents the average, and the white whisker plots the median and interquartile ranges. So each of the purple dots you can see is one fish genome assembly. And along the y-axis, you'll notice it's not alphabetical. These are actually in evolutionary order. So at the bottom here, we've got the oldest taxa reducing in evolutionary time as we go upwards. So here, if we actually look at the content, we're looking at heterozygosity as a percentage. As you can see, there is quite a lot of variability even within each order, but there are some standouts such as siluriforms on the bottom here that we can see are quite distinct. When we looked at repetitive regions, which is the percentage of the genome that is covered in repeats, we can again see that they are quite varied, but also there's a lot of within order variability. There's still a lot of data being generated for this, so we don't quite have everything here yet to see the full picture. When we looked at the transition to transversion ratio, which provides insight into the nucleotide substitution patterns, we can see again that there is quite a bit of variability between the orders. So generally, if genomic changes were considered, genomic changes at the base level were considered to be random, we would expect a ratio of around 0.5. In a biological context, people generally expect to see a ratio between 0.5 and 3, which is where our data has fallen. When you have more transversions, it can be evidence of selection. So the orders that we're seeing here with an average of less than 1 are likely undergoing some form of selection pressure. The next thing we wanted to look at was whether we could correlate phylogeny and ecology with conservation metrics. So here we started by looking at the climate risk index generated by Boyce et al. In 2022. The higher the index, the more at risk the species was considered to be, and they developed this matrix metrics based on 12 different parameters. So here we can see that some of our orders are currently lacking data, but there are quite a few that are higher than average in the vulnerability rating. One metric that could be impacting the climate vulnerability of fishes is the inbreeding coefficient, which we can measure as the percentage of the genome that is made up of runs of homozygosity. So here we can see that generally marine rayfin fish have runs of homozygosity percentage of less than 1, which is quite low. But again, there is a little bit of variability between and within the orders. We generated this plot to have a look at how the climate vulnerability index could be correlated with the runs of homozygosity. And we have actually started to see a trend that's opposite to what we'd expect. Although, as you can see, the R-squared value is near 0, so it's not too significant. What we'd expect to see here is as a fish gets more runs of homozygosity in its genome, it's likely more inbred, and thus the climate vulnerability we would expect to increase. Hopefully, as we start to get some more data into this, we can see if we start to get more of a distinct pattern. So the last thing that our study wanted to look at was whether gene family diversification correlated in any way with species diversity. So this is kind of a work in progress and kind of a what's next. So at this stage, what we've done is we're starting to annotate our genomes using Gemoma. So we're using 5 NCBI RefSeq genomes as references. And so far we've generated predicted protein and predicted annotation files for about 370 of our fish. What we plan to do is What we plan to do with this data is to combine everything and run it through OrthoFinder to identify orthologous genes, protein domains, and architectures. And then we'll be using this data along with the fish tree of life phylogeny as a backbone to generate a cafe analyses. Here we're hoping that we'll be able to identify gene families that have been gained or lost within key fish orders to try and discern some of the evolutionary basis behind them. So we are currently trying to upscale this to quite a large number of fish genomes. So if anyone has any suggestions on this workflow, please do get in touch. And finally, just to acknowledge that this has been a huge team effort. So here I've included the core Ocean Genomes team and of course the partners and collaborators that we've been working with to make all of this research possible. Thank you.
Great, thanks for that talk, Lara. So just a reminder, if anyone has any questions, to pop them into the Slido. We have about 5 minutes for questions now. If anyone has any for Lara, that would be great. Just something to start off, I guess, is you mentioned that you're having difficulty collecting some of the samples, which I can imagine is quite a problem in marine fishes. How do you go about collecting samples or trying to ensure they're high quality?
Yeah, so I think sample collection is quite an issue for most people working in genomics. Marine fishes, I don't think, are too much more difficult than everything else. we've been really lucky in that we have been working with a lot of collaborators to provide us with fresh tissues. So we are trying to sample straight into liquid nitrogen. But when the sample quality is not sufficient, that's when we're still trying to generate these draft genome resources so that there's some representation for all species that we can get our hands on.
Cool. Maybe one other thing is with the repeat analysis that you did, were you able to classify most of the repeats into their different types, or were they mostly like unknown repeats? Like I know from working on other orders like amphibians, most of them end up being unclassified repeats.
Yeah, so for these samples, the repeat analysis I've done is pretty crude. I haven't been looking too much into repeat modeling and repeat masking them fully and properly because I have such a large number of contigs that actually the compute is just, it's outrageous to try and get it done. So if you do have a suggestion on trying to use it on this kind of data, I'm all ears, but it's been quite limiting just the number of contigs to try and run it through that kind of analysis.
Yeah, I can imagine something most people struggle with is compute for large analyses like these. And maybe one more, did you think that using only the short read genomes might be impacting some of your runs of homozygosity analysis? Do you think that findings might be slightly different if you were able to have the long read or high quality genomes available?
Yeah, I definitely think it's something that we need to consider. I have been reading a few papers that have used short read genomes for this kind of analyses, but they've mapped them back to a reference of the same species. Given the number of fishes that I'm trying to work with, that's not feasible. So it's definitely a limitation that I'm trying to work around. Moving forward into the future, it'd be really nice to look at a subset of high quality ones to see if the statistics do hold true. I'm just not sure how feasible it is right now with what we've got. It's quite a large data set and the high quality samples are just not available for a lot of them.
Yeah, for sure. Well, if there's no other questions, we might move on to the next talk then. And thanks again, Laura. All right.
Hello to everyone. Thank you so much for tuning in to my talk today. My name's Tom. I'm a postdoc at the Biodiversity Research Center at Academia Sinica here in Taipei. And today I want to talk to you about what I've been doing in the first 18 months of my postdoc, which is studying chromosome rearrangements in animals. So I'll get straight into it. I'll start quickly with some background. I think one of the most remarkable things that's come out of our ability to sequence genomes to chromosome level in the last 5 to 10 years has been the discovery that really, really distantly related groups of animals share very, very similar genomes with very highly conserved structures. So this seminal paper from 2022 studied genome structure from everything from sponges to cnidarians, and then even to bilaterians like Amphioxus and protostomes like the scallop, and found that the same genes or orthologous genes are kept on the same chromosomes together with really surprising fidelity. So I'll be showing a lot of plots like this today. So just briefly, on these synteny plots, each row is a genome. Each of these black bars represents a chromosome. And then the vertical lines connect orthologous genes, and they're colored by the ancestral linkage group. That is, they're the genes that are the same color were on the same chromosome in the putative ancestor of the species shown on the plot. And what this plot shows is that these very, very distantly related species that probably diverged over 700 million years ago have got the same genes on, many of the same genes on the same chromosome. So this chromosome, for example, is perfectly conserved. And then there's occasional things like chromosome fusions here, here. There's some lineage-specific chromosome fusion, but largely these species have basically the same sets of chromosomes. And from this, people inferred that the last common ancestor of bilaterians probably had 24 chromosomes, 24 of these linkage groups. Now, you could still see almost perfectly in things like the sea cucumber today still has 22 of these intact with just one chromosome fusion here. But this genome structure is quite an evolvable trait, so you can have things like chromosome fusion, chromosome fission, or even translocation, where bits of chromosomes, bits of non-homologous chromosomes switch, that can cause divergences to this ancestral genome structure. And why is this significant? Why do I think this is worth researching? Well, chromosome rearrangement can affect lots of different biological phenomena, from recombination rates, they can cause disease, they might be important in adaptation, they're definitely important in speciation. So you can see over here on the right that that genera with a higher rate of chromosomal evolution also have a higher speciation rate. And for me, I'm just fundamentally really interested in the processes by which genomes evolve. Why is genome structure so highly conserved? The truth is we don't really know. We don't have a good answer to this yet. It might be something to do with gene regulation. These genes need to be kept on the same chromosome because of some sort of regulatory restriction. And it might be to do with something like reproductive sterility, to do with chromosome segregation and meiosis, where there could be problems if there are interchromosomal rearrangements. But the truth is we don't have a really good answer to this yet. So questions that I'll address for you today is I've shown you just a few examples there from very, very few species, but I was interested in how this genome structure evolves more broadly and in less well-studied phyla. Also, when chromosome rearrangements occur, I was interested in the manner in which they happen. And finally, maybe for me, the most interesting thing is what does a normal animal genome look like? If we sequence a completely new phylum of animal tomorrow, do we expect it to have that very conserved genome structure? So I'll start now with the bryozoans. So bryozoans are a group of aquatic invertebrates. They fall within the Spiralia, and they form these colonies of zooids that feed with these ciliated tentacle structures called lophophores. They're super, super abundant. They're found basically everywhere across the world, and they're really important ecosystem engineers because they have these calcified skeletons. Bryozoans fall into one of three classes. Most of them fall into this green class here, Gymnolaemata, but there's two other smaller classes as well. For my project, I had 5 genomes available to me, 4 of them from this green class and 1 of them from this outgroup class here. So what I wanted to do was just look at the genome structure of bryozoans and try and understand how it's related to those of other bilaterian animals. Oh, just to say, these things are super, super morphologically diverse. So this is a plot similar to the one I showed you to start with 3 relatively conserved bilaterians along the top, and then my 4 bryozoans here from that green class. And where you can see those 3 species along the top, we have these columns of genes from the same color that are relatively conserved with occasional chromosome fusion events. In bryozoans, what we found was many, in fact, 18 of the 24 bilaterian ancestral groups of genes are found together on these 4 chromosomes here. And there are some other chromosome fusions and fissions as well. So bryozoans, it seems, or this group of bryozoans has got a very, very, very different genome structure to those other bilaterians we looked at to start with. Or actually, it wasn't just bilaterians, that was all animals, but particularly to bilaterians. Even more surprisingly, when we looked at that outgroup bryozoan, we found again that its genome was very, very shuffled with respect to not only to other bilaterians, but also to the bryozoans as well, also to that bigger group of bryozoans. We did just check very, very quickly that this isn't due to whole genome duplication, and we found no evidence that whole genome duplication had been a factor in this. So from this, we've learned that this phylum of animals, bryozoans, seems to have a different processes governing its genome evolution to maybe what we might have expected. Next, we only have 5 genomes, and I wanted a bigger data set in order to study this sort of more completely. And so I turned to annelids. I think annelids need less introduction than bryozoans. But one thing I think is important to say is that most annelids fall into one of 2 classes, sedentaria, or one of 2 classes, orders, something like that, sedentaria or Erantia. And for this For this, I had 23 genomes, so a much bigger data set. 2 of them outgroups here, and then the rest of them within Erantia or Sedentaria. Importantly, we had 7 genomes from earthworms and leeches, which form this group called Clitolata, which is a subset of Sedentaria. And when I looked at annelid genome structure, most of what I found corresponded very similarly to what the plot I showed you at the start, with largely blocks of ancestral linkage group genes kept together with lineage-specific chromosome fusion events. So this is Erantia on the left and Sedentaria on the right, and largely we can still see these blocks of bilaterian genes kept together in most of these species, again with some chromosome fusions. What was very surprising to us was when we looked at clitolates, that group containing leeches and earthworms, we found their genomes were completely different, like totally shuffled, as if someone had, if the genes were cards in a deck of cards, as if someone had dropped it on the floor and picked it back up in a completely random order. Literally every single bilaterial linkage group is found on every single chromosome in leeches and in earthworms. And again, even between leeches and earthworms, there's been a lot of genome shuffling as well. This is quite interesting because clitolates are quite unusual annelids. They have really, really different morphology to other annelids. They completely lost the larval stage. They have a different mode of reproduction. And they are the group of annelids that has changed from marine to freshwater and terrestrial environments. So there's no good evidence for this yet, but it's very, very interesting that there's a coincidence of this restructuring of the genome and these adaptations to new environments. Okay. Finally, so at this point, I've looked in 2 phyla and found things that I wasn't really expecting. I found genomes that are very, very highly divergent from that structure that I showed you at the start that's conserved not only between bilaterians, but even to sponges and cnidarians. So I wanted to look more broadly. And the reason that those discoveries surprised me was because the general sort of consensus has been that what I showed you at the start is kind of the norm and the expectation, that most groups of animals have largely conserved these blocks of genes with a few chromosome fusion events that are lineage specific in each group. So I got as wide a data set I could find. We're a little bit restricted because you need chromosome level genomes to do this, but I managed to get a data set with 15 bilaterian phyla, and I think over 50 classes. So maximal diversity. And again, just doing these synteny plots, I managed to find species in almost every phylum that had highly shuffled genomes. So genomes that are very, very different where those linkage groups have been lost. There are some phyla, like for example, hemichordates, where all the samples that we had, they have these conserved blocks of genes, like I showed you at the start. But in many other phyla, in nematodes, in tardigrades, in arthropods, and bryozoans, like I already showed you, in mollusks, wherever you look, you can find species with highly shuffled genomes that actually do away with that genome structure. We came up with a way of quantifying this and very, very crudely just classified genomes into high rearrangement or low rearrangement. So pink ones are more shuffled and blue ones are less shuffled, and just plotted these on a phylogeny and did some ancestral state reconstruction. And what this showed was very, very clearly that the conserved bilaterian genome structure has been lost on very, very many occasions, repeatedly in the course of bilaterian evolution. So actually most species, or lots and lots of species, do not have this conserved genome structure. Just finally, when we looked at what this correlates with, it correlates really well with the amount of protein sequence evolution. So genomes that have very, very fast evolving protein sequences invariably also have high levels of interchromosomal rearrangements. Take home messages. Some animals have got incredibly highly conserved genome structure. This can be lost in whole phyla or in specific groups. Can be lost gradually or rapidly. And contrary to previous expectations, actually this conservation of genome structure is probably the exception rather than the rule. That's me done. Thank you very much for listening. Thank you to all my lab members. And thank you to Academia Sinica for funding me. If you're interested in this work, the annelid stuff is out now in MBE, and you can find it by scanning this QR code here. Thanks again for listening.
Great. Thank you so much, Tom. What a fantastic talk. We do have one question in the Slido chat from Rich Edwards, where he's asked, is there any correlation between chromosome structure and the underlying genetic machinery for centromeres and telomeres, etc., e.g. centromere organization or telomere repeat sequences?
Yeah, that's a very good question, actually, and it's something we have thought about, but not sort of done a comprehensive analysis. So it's thought in general that a species with monocentric chromosomes, so a species with one central chromosome, one central centromere, those chromosomes are easier to rearrange because you can sort of just split halves off. They might fission or fuse more easily. So that's a possibility, but actually, like, looking more broadly, I'm not convinced that that is sufficient to explain the differences we see. So, for example, bryozoans and annelids have quite different centromere types, and they both show this high rearrangement. So, although I think it might be important to a limited extent, I don't think it's like the main factor explaining the big differences we see more broadly. The repeat thing is also very interesting. Telomeres I haven't actually looked at all. That's a really, really interesting idea. That's definitely something we can do in the future, especially now with these super high quality genomes. But yeah, so we've characterized repeats in all these species, and I did also, just in not specifically at centromeres or telomeres, but I did look at, you know, that plot I showed that was like rate of protein sequence evolution versus rearrangement level. I did the same thing for repeat content versus rearrangement level, but there was no correlation at all in those things. But I wonder actually if the problem, like looking at repeats very broadly, just looking at all transposons is actually not that useful, and you probably need to look more specifically at different classes or types of transposons. Maybe if you looked at just DNA transposons or just lines or something, that you might see more of a pattern. But that's definitely something we could look at more in the future. Yeah.
Great. I think just following on, like, from a follow-up here from Rich. Are there different patterns in the high C contact maps? DNA Zoo did an interesting study of this in vertebrates, I think.
Ooh, that is an excellent question, and I have to say, not something I've ever considered, but I now will. And yes, that's a really, really good idea. I honestly don't know. But we have high C contact maps for all these groups, so I can have a look. Yes. Rich, could you put your email in the chat or something, and I'll have a look at that and get back to you and say thank you if I if I come up with anything from that, because that's actually a really nice idea. But yeah, at the moment I don't have an answer. Thank you.
Great. I have a question which may be very high level, but these kind of large genome rearrangements, do you think it has something to do with species that have really high or extreme selective pressures? So like those that may be living in more extreme environments?
Yeah, possibly. I think, like, the truth is that nobody knows at the moment. We're only just starting to describe these things just sort of on a very descriptive level. So in terms of actually studying why they're happening, we're really, really not there yet. I think that's very, very possible. One of the reasons we were interested in the clitollates is because they do have this very, very clear difference in lifestyle versus the other annelids. But that's kind of just like a very simplistic, you know, you could, it's a bit of a just so story. It's a bit like, oh, we see this divergence in lifestyle and their genomes are different. Maybe it's a causative thing. The truth is, honestly, we don't have an answer at the moment, but I think the way we'll get to an answer for that is by, in the next few years, looking at really, really broad data sets and starting to build the wider scale comparisons. I think it's very possible that this sort of thing can be adaptive. I think certainly that system of having really, really highly conserved synteny must be adaptive, otherwise there's not a chance it would be kept for whatever call it what you like, 500 million years, something like that. Whether the loss of it is because either it's just suddenly not necessary anymore and it's just like, okay, this doesn't matter anymore, so we can lose it and there's no negative consequences, or whether it's actually something that rearrangement is actually positively selective and adaptive is an open question. But I think it's very possible. Yeah, I think it's a nice idea.
Definitely. Well, that's really interesting. Thank you, Tom. I think we haven't got any more questions in Slido, so I think we'll let you go and
Sure.
probably
Thanks
wrap
so
up
much.
this session. Thank you.
Thank you.